Hello, my dear viewers. Ever thought about what makes up your body? Just like bricks build a house, it's teeny powerhouses called cells, the invisible builders of our life. From healing cuts to powering your brain, these little wonders do it all. In this chapter, we will uncover how cells were discovered and what they are made of, how plant and animal cells differ to each other, and what they rightly called the unit of life. Get ready to zoom into the microscopic world where all life begins. What is a cell? Basically, the cell is the fundamental structural and functional unit of life. Every part of a living organism, that is, or organs in animals or parts of plants, is made up of cells. All life of all living organisms starts from a single cell, usually an egg. Cells are microscopic structure and cannot be observed under the naked eye with the naked eye, hence the invention of microscopes. The invention of microscope and the discovery of the cell, Robert Hooke, the English scientist basically, who invented a compound microscope used with two lenses, first observed cork cells and coined the term cell. This is the cell, and this is the Hooke's compound microscope setup. It is the light source. Oil lamp is there. Flask filled with water to focus light. There is a mirror and lenses. This is Hooke's drawing of cork cells, which resembled tiny boxes or rooms, leading the term cell. Hooke firstly proposed the word cell. Robert Hooke's observation. Hooke observed a thin slice of cork using under his microscope. He noted it was composed of many tiny box-like compartments, which reminded him of the rooms or chambers or cells in a monastery. He coined the term cell based on these empty compartments. The cork cells Hooke saw were dead cells only showing cell walls. Later, Antony von Leeuwenhoek, a Dutch scientist and lens maker, created the first microscope using a single biconvex lens. His microscopes were simple yet powerful enough to observe microscopic life like bacteria and protozoa. Compound microscope, an advanced version of Hooke's original microscope. Key parts include it included eyepiece, objective lenses, low and high power objective lenses are there. This is the stage, mirror, this is arm, this is base, coarse and fine adjustment, knobs used for focusing the image, body tubes and nose pipe hold and rotate lenses for better viewing. It uses glass lenses and light to magnify up to 2000 times. Electron microscope. It provides magnification up to 2 lakh times. Beams of electrons are used instead of light. Lenses are replaced by magnetic fields to bend electron beams. It offers for greater resolution and detail compared to compound microscopes. It enabled discovery of fine cell structures, organelles, membranes, etc. Cell theory contributions. Matthias Leiden in 1838 proposed that all plant cells are made up of cells. Theodor Schwann proposed that all animals are made of cells. Together they proposed the cell theory. Rudolf Virchow in 1858 added that all cells arise from pre-existing cells. The cell theory states that three major points. The cell is the smallest unit of structure of all living things. The cell is the unit of function of all living things. All cells arise from pre-existing. This is the image of the electron microscope, which the magnetic lenses are used. Electromagnetic lenses, it offers greater resolution and detail compared to the compound microscope. Every organism like mango plant or frog animal is made up of cells. Cells are basically structural units. Every part, leaf, limb, etc. shows a cellular structure. Functional unit. Every function like digestion, movement, photosynthesis is driven by specific cells and their components. Now we should know cells. How many cells are there and how small the cells are. Cells die and are replaced. Our body, like that of a frog or a mango tree, is made up of millions of cells. Old cells die and new ones form from pre-existing cells. This is a continuous cycle. All life starts as a single cell. Every organism begins life from a single cell. A frog starts from an egg cell. A mango tree starts from a seed that develops from a single cell in the ovary of the flower. Cells, how numerous? Larger the organism, more the number of cells. Single cell organisms made up of just one cell, examples bacteria, yeast, amoeba. Few celled organisms, they have only a hundred of thousand cells. Examples: Spirogyra, Volvox. Multi-celled organisms made up of millions to billions of cells. Example: human, mango trees, etc. Human body cell facts: 1,000 million or 1 trillion cells in the body, 10 billion nerve cells in the brain, 5 to 6 million red blood cells, and 7,000 white million blood cells per cubic millimeter of blood. Cells. How small? Cells are microscopic and vary in size. Smallest bacteria or red blood cells, longest হচ্ছে nerve cells, it up to 1 meter in length. Largest bird egg like ostrich egg, it is visible to the naked eye. Why are cells small? The efficiency of small size. Cells stay small for two major reasons: faster communication between cell parts, greater surface area to volume ratio, allowing it efficient exchange of gases, oxygen, CO2, nutrients, and wastes. When a large cube is split into smaller tubes, the total surface area doubles. More surface area means faster nutrient and gas exchange, better waste removal, quick response to damage, etc. Think of it like this: smaller cells, more efficient life processes. Cell shapes. To suit functional requirements, cells have different shapes based on the function they perform. These shapes include disc-like cells, polygonal, rectangular, cuboidal, thread-like, branched, irregular, etc. These are the different kinds of cells of different shapes. Epithelial cells are flat or polygonal. They provide the surfaces, provide protection. Red blood cells are circular and biconcave. They transport oxygen through narrow capillaries. White blood cells are amoeboid in nature. They squeeze through capillaries and fight against infections. Cartilage cells, they are rounded and smooth. They provide cushioning and supporting in joints. Connective tissue cells, they are irregular and web-like. They provide structural support and connection. Striated muscle fiber, they are long, cylinder and striated. They help in voluntary movement. Bone cells, they are long extensions are there. They are help in support and communication. Smooth muscle cells, they are spindle shaped in nature. They are involved in involuntary movements like the digestion. Nerve cells, they are very long with branched end, transmit electrical impulses. Chlamydomonas, they are round with flagella. Their movement single celled organism. Plant cell from leaf, rectangular with thick wall. Photosynthesis is structure, it provides structural support. Guard cells of stomata, it is bean shaped, and open and closed stomata or pores are there. Amoeba, irregular and shape changing movement, and ingestion via pseudopodia. Functional notes: RBCs are circular and biconcave. They help move through narrow capillaries. WBCs are help in amoeboid movement. They help them squeeze through tissues. Nerve cells, they are long and conduct impulses over long distances. Muscle cells, they are long and contractile, help in pulling movement. Guard cells are bean shaped and control the opening of stomata in leaves. Structure of a cell: all cells have three essential parts: cell membrane, outer boundary, nucleus, it control activities. Cytoplasm, jelly-like substance where cell organelles are found. Cell organelles, they are specialized structures within a cell that perform specific tasks similar to organs in the body. These are vital for the cell's function and survival. These are the images of the plant cell and animal cell. You can find out the differences between the two types of cells. Parts of a cell: living parts are cell membrane and non-living parts are cell wall, which are found only in plant cells. In the cytoplasm, the parts of a cell are endoplasmic reticulum, mitochondria, Golgi bodies, Golgi apparatus, ribosomes, lysosomes, centrosomes, which are only found in animal cells, and plastid only found in plant cells. Granules, vacuoles, food fat droplets are the non-living parts found in the cytoplasm. The living component of the nucleus is the nuclear membrane, nucleoli, and chromatin fiber, and the non-living parts are the nucleoplasm. The cotton, jute, coconut fibers are made up of cell walls from dead plant cells. Cell membrane and cell wall. Cell membrane are the plasma membrane. They present in all cells. They are selectively permeable, allow only certain substances to pass in or out. Cell wall, it is present only in plant cells. They are made up of cellulose, mainly cellulose. It is a non-living component. It provides rigidity, shape, and support. Allow free movement of substances. It is freely permeable. Endoplasmic reticulum. Smooth endoplasmic reticulum lacks ribosome involved in lipid synthesis and detoxification. Rough endoplasmic reticulum has ribosomes aids in protein synthesis, connected to the nuclear membrane and act as a supporting framework for material transport. Ribosomes are the sites for protein synthesis. The tiny granules in the cytoplasms are attached to rough endoplasmic reticulum. Factories for protein synthesis. Ribosomes are the factories for protein synthesis. Mitochondria, they are basically the powerhouse of the cell. This is the site of aerobic respiration. Double membrane structure with cristae. This is the inner force. It produces ATP, the energy currency of the cell. Golgi apparatus, it is the delivery system. It modifies, stores, package proteins and lipids, appear as stacks of membrane and vesicles near the nucleus, involved in the secretion of enzymes, hormone, etc. Lysosome, intracellular digestion, they contain digestive enzymes, they destroy and digest foreign substances, or also digest worn out cell parts known as suicide bags. They are found only in animal cells. Centrosome and centrioles, they are found in animal cells, helps in cell division by organizing spindle fiber. Plastids, they are found only in plant cells. Chloroplasts or green plastids carry out photosynthesis. They vary in shape, oval, spherical or disc-shaped. Plastids are basically double membraned organelles and found in plant cells. They are classified into leucoplast, chromoplast and chloroplast. These are the difference between plant cell and animal cell. A plant cell have cell wall outside the plasma membrane, and animal cell the cell wall is absent. Plastids are present in plant cell, but plastid is absent in animal cell. Vacuoles are present in plant cell. It is single and large central vacuole is present in plant cell, but in animal cell many small vacuoles are there. Centriole absent, and in case of animal cell centriole is present. Nucleus, it is generally eccentric, Mitochondria is less in number and more in number. Golgi body, it is many and scattered. Animal cell, it is single and located near the nucleus. Leucoplast, leuco means white, colorless plastids, no pigment present there. They are specialized for storage like starch, fat, proteins, etc. They are commonly found in potato tubers. Chromoplast, chromo means color. It contains color pigments like xanthophyll, yellow colored pigment, carotene, orange red pigment. They are present in petals and fruits. They provide attractive colors to aid in pollination and seed dispersal. Pigments like blue, violet and purple are not stored in plastid but dissolved in cell sap and are called as anthocyanin. Chloroplast, chloro means green. The green plastids contain chlorophyll. They perform photosynthesis. They can convert light energy into chemical energy like glucose. They use carbon dioxide and H2O to synthesize sugar and starch. They also contain DNA and can divide. Green turns into red. Example of raw tomatoes and chilies are green due to chlorophyll. As they ripen, chlorophyll breaks down and the carotene becomes visible. Nucleus, the control center of the cell. Its key function, it regulates and coordinates cell processes. It controls cell division, contains gene for heredity. Structure, nuclear membrane separates the nucleus from the cell. Nucleoplasm, it is the fluid substances inside the nucleus. Chromatin fibers, the thread-like structure within DNA. During cell division, chromatin condenses into chromosomes. Types of cells by nucleus. Prokaryotic cells have no nuclear membrane, example bacteria. Eukaryotic cells have a well-defined nucleus like plant and animal cells. Chromosomes are hereditary carrier made up of chromatin, which contains DNA and genes. Genes control traits that are passed from parents to offspring. These are the chromosome numbers in various organisms. Human have 46 chromosome or 23 pairs of chromosome. Ascaris, round off have 2 chromosome. Garden pea has 14 chromosomes. Onion have 16 chromosomes, and like that. Key points: chromosomes are hereditary carriers made up of chromatin, which contain DNA or deoxyribonucleic acid and genes. Genes control traits and are passed from parents to offspring. Non-living substances or cellular inclusions. They are granules, tiny particles storing starch, granule, glycogen or fat. Fluid filled sacs are present in the cytoplasm. Plant cells have large vacuoles with cell sap, store sugar, salt, amino acids. Animal cells have smaller or no vacuoles are there. DNA fingerprinting. DNA fingerprinting is used for identifying individuals like fingerprints. The paternity testing, criminal investigations. Example, in 1995 in a Delhi murder case, DNA from a girl was matched with her parents' blood cells to confirm identity. Gene versus chromosome. It's not the number of chromosomes but the type of genes they carry that determine the characteristics. Example, a lion, tiger and a house cat all have 38 chromosomes but look different due to different genes. These are the table containing cell organelles, their main characteristics and function. Their plasma membrane, it separates cell from the surrounding. It regulates entry and exit of substances, maintains the shape in animal cells. Cell wall, it has non-rigid structure and it gives rigidity and shape to the cell. It allows solute to exchange freely. It provides mechanical support. Cytoplasm, present between plasma membrane and nucleus, mixture of water, salt and organelles. Their site for metabolic reactions, hold organelles, their site of respiration. Endoplasmic reticulum, it forms a tubular network, supportive framework, synthesis of transport of proteins and fats. Mitochondria, sausage shaped structure, inner folded membrane. Cristae are there, they have DNA and ribosomes. The powerhouses of the cell releases ATP via aerobic respiration, synthesis of respiratory enzymes. Golgi apparatus, they stack of flattened membrane sacs, contains tubules, vesicles or vacuoles. And the synthesis and secretion of enzymes, hormones, they form acrosome of sperm. Ribosomes, the small granules in cytoplasm or endoplasmic reticulum, they are single walled and composed of RNA. They are the site of protein synthesis. Lysosome, the membranous sacs from Golgi bodies. They contain 40 different kinds of enzymes. They help in intercellular digestion, destroy foreign substances, act as suicide bag in injured cells. They help in digestion of bone and cartilage. Centrosome, plastid, nucleus, nucleolus, chromatin fibers, vacuoles, granules, etc. Keynotes: Golgi apparatus is like the cell's post office. It's responsible for modifying and packaging proteins and lipids. Ribosomes and nucleolus work closely in protein synthesis. Lysosomes play a key role in cell defense and waste removal. Chloroplasts are vital for photosynthesis. While chromoplast and leucoplast have coloring and storage roles respectively. Nucleus is the control center and chromatin carries the genetic code. Differences between plant cell and animal cells: we know the cell wall is present in plant cell, it is absent in animal cells. Centrosome, it is absent in plant cells but present in animal cells. Vacuoles, large and prominent in plant cells, small and temporary in animal cells. Plastid, present in plant cells but absent in animal cells. Cytoplasm is less dense in plant cells. Animal cell, it is dense and granular. Cytoplasmic arrangement is thin lining but it fills the entire cell in the case of animals. Protoplasm is the living substance inside cells. It contains both cytoplasm and nucleus. It is translucent fluid, colorless to greyish or brownish. It includes water, protein, carbohydrate, fats, mineral salts, etc. It is considered the total living matter in a cell. Protoplasm is equal to cytoplasm plus nucleus. The cell is the basic structural and functional unit of life. It is enclosed by a cell membrane with or without nuclear membrane. Plant or animal cells have a nucleus and well-defined membrane bound structures. Prokaryotic and eukaryotic cells, they are primitive with a true nucleus. They are prokaryotic cells like bacteria or cyanobacteria, and eukaryotic cells are advanced cells with a true nucleus and membrane bound different cellular organelles like amoeba, animals, plants, etc. These are the difference between prokaryotic and eukaryotic cells. We know that every activity of a living organism is the outcome of cellular activity like growth and development, repair, movement, feeding and nutrition, circulation of blood, respiratory gas transport, body protection, sensory perception, thermoregulation, reproduction, water absorption in plants, photosynthesis, flower attraction, inheritance, etc. So the summary statement is that there is not a single activity in the body of an organism which is not carried out by the cells, but cells of course are specialized for the particular functions. This is the points to remember. Thank you my dear students, all the best for your studies. Stay focused and keep shining. If you found this class helpful, don't forget to like, share and subscribe for more. Drop your questions or feedback in the comments below. See you in the next video. Until then, take care and keep learning.